drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. John, Blenis, thank you guys for joining me.
Thank you for having us.
Yes. This is the first. I've had a couple dual interviews before, but usually they're separate channels, and you guys are there in the room together. Do you guys want to just take, I don't know, one or both of you just take a moment to introduce who you guys are and where you're calling in from and what the lab does?
Sure. Yeah. So my name is Nikos Koundouros. I'm a postdoctoral fellow in John's group here at the Meyer Cancer Center at Weill Cornell Medicine. And really my research interests since I've joined John's lab is understanding the fundamentals of nutrient sensing in breast cancer. And really this work has led to some interesting findings as they relate to dietary fat intake.
Hi, my name is John Blenis. I'm a professor of pharmacology. I'm the associate director of basic science for the Meyer Cancer Center. And I'm also the Anna Maria and Stephan Kellen Professor of Cancer Research here at Weill Cornell Medicine.
Yeah, so you guys have had some interesting work come out recently that links components of diet to at least one type of breast cancer. And to me, it's really interesting. It's related to a lot of things I've been reading about and learning about recently, both on the podcast and elsewhere. But I want to give people a sense for some of the a basic sense for some of the things that will be important for understanding the recent work that you guys have published. And I want to start with mTOR. And so we'll start pretty basic, but a lot of people listening will have some familiarity here. But starting just bare bones basic stuff here, what is mTOR? What is it doing inside of cells at a very high level?
Well, actually, I'd like to start a little bit in the past
Hmm.
because I want to illustrate that the studies on mTOR originated from studies in yeast. In 1993, there's a paper published in Science from Michael Hall's lab, sorry, 91. And then in 92, in the 91 paper, he found that this drug, this natural product called rapamycin, very potently would block the growth of yeast. And so he used that model in yeast to identify the target of rapamycin. So rapamycin is this bacterial macrolide that was isolated from bacteria from Easter Island. And then he identified the target genetically and called it target of rapamycin, or TOR. And now it's referred to as mechanistic target of rapamycin. And then the following year, we published a paper showing that rapamycin would block the activation of an enzyme called Essex kinase. And that was in mammalian cells, so suggesting that the system was working in mammalian cells. And then a couple of years later, we showed that the target of rapamycin was acting between another really important enzyme called PI3 kinase, which is mutated in many, many cancers, and the Essex kinase that we had identified. And since then, over the years, the mechanisms of how TOR is regulated have been defined, and it's extremely complex. And this enzyme acts like a brain. It's like the brain of the cell. It can sense the availability of nutrients, of growth factors, of stress, oxygen status, energy status. It basically scans its environment and then determines, do I have enough energy and building blocks and things to then make things through these anabolic processes that are important for cell growth. And when improperly regulated by activation of proto-oncogenes or loss of tumor suppressors, then this inappropriately takes all these nutrients and makes the building blocks for cells to grow in an uncontrolled fashion. So the basic concept is that TOR is a really amazing signaling system that is a sensor for the cell, is the brain of the cell.
So it's a sensor. It can sense aspects of stress, aspects of nutrient availability. It's like an integrator of many signals, and it's very much involved with a cell's sort of decision about when or how much to build new stuff.
Exactly.
And so this brings us to the basic concept in cell biology or biochemistry of anabolism versus catabolism. Can you just explain what that means for people and how that hooks into mTOR?
Sure. So, I mean, at a fundamental level, so anabolism is all about building things and synthesizing biomolecules and macromolecules. So TOR is very much involved in promoting anabolism and these kind of building processes. So when it's active, it leads to increased protein synthesis, increased lipid metabolism, so cells being able to synthesize fatty acids and more complex lipid species de novo. It's also involved in nucleotide synthesis. And then you contrast that with catabolic processes. So this is largely, breaking down biomolecules in the cell and kind of facilitating recycling processes. So autophagy kind of comes up as the main pathway involved in this. And so it's accepted that, TOR is a negative regulator of autophagy. So in this way, it maintains the balance between anabolism and catabolism in that context.
Mm-hmm. And so basically, at the cell biological level, we often could just think of all of the reactions as either being anabolic or catabolic. A cell has to either build things up or break things down, depending on what's available in the environment and what's going on. You might want to build a bigger synapse or a new synapse in the brain. You might want to tear one down. And both are important in different contexts. You might want to make your muscles bigger. You might need to have your muscles atrophy so you can use them to create energy for something else. So cells are always either building up or breaking down, and there are some exquisite systems in the cells that allow it to integrate a lot of different signals in order to make that decision. And obviously that decision needs to be highly regulated, and it needs to be done in a appropriate fashion. And TOR is really important for that. And TOR is really an anabolic on the anabolic side in the sense that if mTOR is activated, it's going to mean sort of growth oriented stuff happens.
Exactly.
Got it. Okay. Well, the other thing I want to make sure people know, so some of these terms that get thrown around. So we know what enzymes are. I'm going to assume that the audience knows enzymes if they're listening to this podcast. But what is a kinase? What type of enzyme is that? And what are those doing?
So a protein kinase is an enzyme that takes a phosphate off of ATP, and then it transfers that phosphate to a target protein. And that event, that phosphorylation event, can modify the protein in terms of its activity or its location or its stability, um, and um, and thus changes the downstream signaling events that are downstream of TOR or the kinase. So the TOR pathway is very interesting because there are multiple kinases that are linked to mTOR. There is the PI3 kinase, which is a lipid kinase, and then there's an enzyme called AKT, which is another kinase that phosphorylates components that suppress TOR. When phosphorylation of that protein occurs, it inhibits its ability to suppress TOR, and then TOR becomes active. And then TOR signals to other protein kinases, and they do their thing. They phosphorylate the other downstream targets. So you have this cascade of events that leads from a signal and then is propagated and amplified to regulate many biological processes, such as the processes that we were talking about, the anabolic processes.
Mm-hmm. And at a very so a couple things. So kinases, they phosphorylate things, and if we think about like ATP, the way that I sort of started to learn when I was a student about phosphorylation and think about how this stuff works is, energy is sort of contained in all of these chemical bonds that get made and broken in the cells. And when things get phosphorylated and dephosphorylated, it's sort of like these bond, energy is getting passed along. And when something's phosphorylated, it's either sort of switched on or moved somewhere. It causes something to happen. It's like the chemical way that the cells sort of pass the torch of energy to regulate what's happening where.
Yeah, it's often referred to as a process called signal transduction, or signal propagation, and and it's really tightly regulated. It's very cool because TOR regulates all these processes that make ATP, which it needs to signal. But when there's not enough nutrients to provide for the ATP, then it's no longer available for mTOR, and therefore you decrease signaling by multiple other very intricate mechanisms.
And so at a high level, I would assume that, if mTOR is this anabolic thing, well, obviously, if there's enough nutrients around, there's a lot of nutrients, whatever exactly that means, that's going to tell TOR, okay, like we've got plenty of stuff. We've got all the things we need for energy. We have what we need to build stuff. And so we can start building stuff. But when we start getting into some of the details here, is there anything specific in terms of, is TOR responding more to fats versus proteins or amino acids? Is it responding more to sugar? Is it a general nutrient sensor, or how much specificity is there in terms of what it's sensing on the nutrient side?
Yeah, so that's an excellent question. So I think at a very high level, a lot of the mechanistic work has focused on kind of glucose and amino acids.
Mm-hmm.
And I think for the large part, a lot of these studies have indicated that, both are potent general activators of TOR when cells are provided with these nutrients. And then kind of more recently, lipids and fats have come into the picture. I think what's a really interesting direction in the field, and maybe less appreciated and needs to be studied a bit more, is whether there are specific signaling nodes downstream of mTOR that each nutrient might differentially regulate.
Mm-hmm. Mm-hmm.
So I think, a lot of assays and a lot of kind of big screens that look at the phosphoproteome of TOR, for instance, mostly look at these nutrients in combination and then kind of probe all the kind of downstream substrates. Whereas, I think a really interesting direction in the field is asking, is glucose regulating a specific substrate, a group of targets of mTOR? Are amino acids doing the same thing, and same with fats? And so I think getting that more complete picture and then understanding how all of those are integrated, as John said, to then really give a clear picture of what the nutrient environment of the cell is an important question.
Mm-hmm. But TOR is not sort of merely reading how much total ATP or potential ATP is there. There's a lot of responsiveness that's probably coming from individual nutrients, and
Exactly.
we haven't completely mapped out all of those intricacies yet.
Exactly. Yeah.
Yeah, we've
Interesting.
mapped out a lot of them, but it's really, again, when you think about the brain, and I know your background is in neuro, um, it's there are so many different signals that are
Hmm.
integrated at that level. And then as Nico said, there could be specific signals downstream, but we do know a lot about how certain amino acids, for example, regulate mTOR. We
Hmm.
know a lot about how things like insulin or growth factors regulate mTOR. We know a little bit less about some other amino acids. We know, and until Nikos' study, we didn't know much about how essential fatty acids could regulate TOR pathway. The, and yet, there's still a lot more to learn.
Hmm. Hmm.
And we're talking about mTOR and the cancer background, but we've already been talking about how nutrients can regulate mTOR. Now, just think about nutrient overload.
Hmm.
So, nutrient overload could be eating too much or eating the wrong things, and that will activate TOR and will activate it inappropriately, and then that can contribute, as Nikos has shown, to either cancer, but also to obesity,
Yeah.
to cardiotoxic events, diabetes, and there's a lot of evidence in other model systems like Drosophila and in flies, Drosophila and worms, and even in yeast, linking toward aging and these metabolic diseases. So there's really a lot we still need to learn about it, um, and this is why it's so important to fund basic science.
Yeah. Yeah. And,
So
I
there's
guess,
a lot to dissect this pathway.
yeah. And when we think about growth and, anabolism, TOR is obviously this really critical integrator, this really critical node here, but there's many ways that you can have healthy growth and many ways you can have unhealthy growth. So even though we will talk largely about cancer here, which would be an example of unhealthy or dysregulated growth, there's many other things that are unregulated or unhealthy growth, like obesity and things like this, that probably tie into TOR as well, almost inevitably. And even though we're not going to talk about them specifically as much, right, like just the basic concept of anabolism, of growth, like it has to be finely regulated because there's many different ways it can go wrong. And TOR, we're gonna end up talking about one already complicated story here, but there's a whole world of complexity here that has only been partially mapped out.
Right.
And so let's talk a little bit about cancer at a basic level. So cancer, at the basic level is just some kind of unregulated or unchecked growth in cell division. But when we think about cancer in general, not necessarily just breast cancer in particular, what are some of the things here that might be relevant to the story that you guys are going to tell us about in terms of how cancer cells go awry when it comes to things like nutrient sensing and nutrient acquisition? I would assume that a cancer cell has to have certain sort of tricks and tools it uses to get more than its fair share of nutrients, and maybe mTOR is behaving in certain characteristic ways in cancer cells. What can you tell us there?
Well, I think, it's very clear already that things like elevated sugars are bad. And what cancer cells can do is they can increase the expression of cell surface transporters that will suck up the glucose, for example,
Mm-hmm.
and actually make the microenvironment depleted of glucose. So therefore, your immune cells will have less glucose availability because the tumor cells are taking it up actively. And there are other examples of this where tumor cells really take advantage of their ability to increase metabolism, to take up metabolites, and that gives them an advantage over the surrounding tissue. And even under conditions where you have low nutrient availability, they're still able to take up whatever's available because of these increased metabolic processes, these increase in transporters. I think that's a general view.
Yeah, and I think just to build on that, I think when also we're thinking of cancer at a high level, it's really fascinating to consider these metabolic adaptations in the context of the underlying drivers and aberrations of disease. Going back to the whole insulin and PI3K signaling aspect, mutations in PIK3CA, for instance, are one of the most frequent genetic events across all human cancers. And this can lead to, for instance, increased insulin sensitivity, but also it can provide a selective advantage to cancer cells. So maybe in nutrient deprived conditions, for instance, it does still provide these pro-growth signals to give the cell essentially enough time to adapt. So I think a big area of cancer research, and certainly the way we're thinking about it is, how are these metabolic networks integrated with these kind of genetic drivers of disease that we're able to kind of diagnose and identify through sequencing, for instance.
Mm-hmm. And I would imagine, on the one hand, there's many different types of cancer, and they're all going to have their own unique aspects, but cancer generally, right, that they have to be able to, these cells have to fuel their growth. There are probably sort of relatively general things that most cancers get good at that allow them to be cancer or become cancer, or allow them to persist and grow as cancer. And one of them that you mentioned has to do with nutrient acquisition. And so some cancers have the highly expressed transporters that allow them to basically just suck up nutrients from the environment more than our cells otherwise would, and therefore they're just sort of just take more of their fair share from the nutrients that are in the body.
Exactly. Yeah.
And so let's go over the breast cancer basics. One of the things, we're going to talk about one form of breast cancer in your study. And so I want people to appreciate the different forms of breast cancer. And the other thing here that this ties into that I think is interesting, that maybe we'll get into a little bit, is like how the different subtypes of cancer and some of the other partitions we can make among people sort of prevents us from seeing things that are nonetheless like true aspects of biology. And so let's just start with the different subtypes of breast cancer. What are the major forms here, and what are their major distinguishing features?
Sure. So breast cancer is a highly heterogeneous disease. I think this was kind of first realized in the early 2000s when kind of sequencing of different breast cancer tumors was done. And so I think at a high level, breast cancers can be divided into hormone receptor positive. So these are cell lines, or these are tumors that express, for instance, the estrogen receptor, progesterone receptor, and then the HER2 receptor. And then within those, you have distinctions into luminal A, luminal B, based on the balance of estrogen and progesterone receptor expression, and then HER2 amplified.
Triple negative,
and then
triple positive.
And then triple positive as well. So tumors that express all three receptors. And then the other kind of subtype it's largely accepted to be the more aggressive subtype. It has an incidence that occurs earlier in, for instance, a woman's life. So diagnoses usually occur in the late 30s, maybe even 40s, and it's associated with more aggressive diseases as triple negative subtype. So these are tumors that lack expression of all hormone receptors. And really what makes this subtype in particular really difficult to treat is that there aren't really any targeted or personalized therapies against this subtype. So when you think in particular of hormone receptor positive and tumors that have estrogen receptor in particular, for instance, there are clear hormone therapies that if a patient has this tumor type, a specific drug can be prescribed, the same for HER2 amplified tumors. But really for triple negative, because they don't have these clearly defined biomarkers, the standard of care at the moment is just chemotherapy. There are some exciting trials coming out looking at, antibody drug conjugates and immunotherapy, but for the moment, really chemotherapy is the first line therapy.
Mm-hmm. Yeah. Okay. So there's different forms of breast cancer. They can be distinguished based on proteins, things like estrogen receptor and other things that the cancer might or might not express. And in the case of the triple negative form of breast cancer, it's defined by the fact that it does not express three different things that other breast cancers do express, and that's tied to the fact that this is a hard one to treat. It can't sense things like estrogen, and therefore it is things like estrogen or other hormone therapies. It's just not amenable for this type of cancer.
Exactly.
Right.
Got it. And do these markers, like, do they tend to have something? Are they kind of just markers that allow us to like distinguish them a little bit, or are they tied into the functionality and the treatment of the different cancers in a close way?
Well, they are markers like HER2 and ERPR, are markers, the lack of those markers in triple negative, which is what defines triple negative. The markers allow for directed therapy. So, for example, if you have a HER2 amplification, there are antibodies that recognize HER2 that you can use to selectively target those HER2 positive cancers. 30 or 20 years ago, those were very deadly. Now, because of these targeted therapies against HER2, the mortality rates are dramatically reduced. For the hormone responsive cancers, estrogen and progesterone, tamoxifen is a long used antagonist of estrogen receptor signaling. There are other ways of targeting breast cancers that have estrogen receptor signaling, like aromatase inhibitors, which block the production of estrogen. And as Nico said, in triple negative, we don't have those sort of markers or targets at this time. Now, I just want to clarify that we say triple negative have no hormone receptors. That's not quite true. There are other hormone receptors that they express that may be down the road therapeutic targets. But right now, they're not.
Yeah, so there may be other hormone receptors. There probably are. We just don't know what they are yet. But the triple negative is it doesn't this version of breast cancer doesn't have the three that we sort of know well that other cancers have.
Right.
Got it. And so based on what you said, it sounds like the triple negative form of breast cancer is one of the, if not the deadliest version, because we just don't really have good ways of treating it yet. The other ones have seen big improvements over the years. They used to be much more deadly than they are today. So we're making progress on some of them in terms of how deadly the cancer is. What can you tell us about how common these different cancers are in terms of how frequently they originate? Are they going down across the board, up across the board? Is it mixed bag? What do we know there?
Well, I think, and you may correct me if I'm wrong, triple negative rates are increasing in the young. And the hormone responsive cancers are increasing in elderly. Now, the link between age and cancer is not absolutely clear now. It's still a major area of investigation. But that increase in incidence could also be because we live longer.
Right.
So there are a lot of these things that have to be taken into account. But I think the fascinating thing and the scary thing is that triple negative breast cancers are starting to occur at a higher rate in younger people.
Yeah, that is interesting and disturbing. And that's true for other cancers, I believe, although I don't know a lot of the details there. But one of the things this makes me think of, which clearly is going to tie into maybe some of what drove your research, but when you see something like a cancer becoming more common specifically in younger people, to me, that starts to get me thinking, okay, what are younger people exposed to in the environment more than their parents and grandparents were? Because, they're obviously inheriting their genes from their ancestors. But if it's a specific problem that's growing in young people, that starts to get me thinking about environmental exposures that might be more common today. And then a big obvious one that's, I think, pretty glaring is our diets are very different than our grandparents' diets, for example, and we're exposed to different types of things, often at higher concentrations and often starting earlier, even in utero. And so, what do we know? Before we get to your study again, there's a lot more I want to cover off on. What are some of the key things just in the realm of the link between diet and cancer that we know about? You mentioned sugar. High sugar can be an issue, but I don't know, what are some of the major things we should talk about in terms of what is already known before your study in terms of how diet in general and specific dietary components link to at least some forms of cancer?
Definitely. So I think to get started on that, so I really think it's within the last 10 years or so that we've had a lot of these high-level population-based observational studies that have been trying to look at the associations between lifestyle habits and the incidence of certain cancers. So I think it's a striking statistic, but I think it's something like close to 50% of all cancers have some type of or can be modified in some way by lifestyle factors and how patients respond to therapies and the overall disease trajectory and relative risk lifestyle plays a major role. So, we can talk about diet, we can also talk about physical activity and excess body weight as a whole.
Hmm.
I think there are some cancer types specifically that have been shown to maybe be more kind of sensitive to these type of lifestyle factors. So, breast cancer is one, for instance, where excess body fat and kind of leading a more kind of sedentary lifestyle can have or can increase the risk for disease. I think as John mentioned, when we think about also the fact that certain cancer types are being diagnosed at early and earlier ages, I think colorectal cancer is one of these cancer types that's showing that concerning trend. And
Yeah.
I think there's more of an effort to understand now about maybe how sugar and sugary drinks as a whole, for instance, high fructose intake, for instance, might contribute to that worrying trend. Um, so yeah, I think at a very high level with these population-based observational studies, they've certainly shown these associations and some very convincing correlations. And I think what's really an exciting frontier now is trying to understand the specific mechanisms about why this is happening at a cellular level, and then trying to understand kind of what biomarkers are specifically responsible or dictating these trends.
Hmm. Hmm. And so, obviously, when we talk about diet and cancer or diet in any disease, diet's a pretty big world. There's lots of different dietary components. People's diets differ in all sorts of ways, and we're going to dig into some of that. But if we just think about this in very, very simple terms, and I just like in a common sense fashion, based on the basics you told us about mTOR and the basics of what cancer is, in general, I think we would just sort of expect, right, that if you're eating too much of anything, not that everything's equal, and we're going to dig into some of the details, but if you're promoting growth too much too often, that's you're promoting anabolic growth generally. And if you're doing that in a way that is, I don't know if I want to say unnatural, or is going to have some probability of dysregulating the biochemical processes that regulate that growth, the more you go in that direction, the more cancer risk you're generally going to have. Is that roughly a fair thing to think?
I think it's safe to kind of draw those conclusions, and I'm sure we'll kind of touch on it touch on it a bit later. But I think when we think about the role between diet and cancer risk as a whole, the points that you raise are completely true. Where maybe overnutrition as a whole, you're leading to these activation of these pro-growth pathways.
Mhm.
But I think it's especially with diet, it's tricky to kind of make blanket recommendations.
Yeah.
And I think maybe this is a kind of an obstacle when we think about how can we integrate nutrition in a cancer patient's journey. I think these kind of blanket recommendations, while they're helpful at a high level of understanding correlations, how we can match
Mhm.
specific patients to the right diet to complement their existing therapeutic regime or improve their quality of life is still a major question.
Mhm. And then I'm not sure exactly how to formulate this, but um, when you think about the structure and sort of the cell biology and the biochemistry side of TOR and what it's doing and sort of the mechanics of it that you guys are experts in, and when you think about I'm trying to get at like the natural dynamics of TOR regulation, whether or not we even know anything about that, given that, but by, we're doing all of our studies in mostly in non-natural situations. Is sort of the TOR system, is there any indication that it's sort of very tunable and modular in terms of its temporal dynamics? Is there any reason to think that maybe it's supposed to sort of go up and down, given sort of the natural waxing and waning of feeding behavior that we see in the natural world as animals, are, going from season to season and, going from sort of feast to famine cycles? Like, is this system sort of meant to always be on, or is it meant in some sense to kind of be on, off? Do you know what I'm saying?
Yeah, I mean, it's as I said before, it's really a sensor of the environment.
Mm-hmm.
So when you don't have access to foods, it'll turn down, and that's important because you don't want it on because it's consuming energy. But
Mm-hmm.
when you reduce the access to nutrients, it actually turns down. When you sleep at night, it turns down.
Mm-hmm.
The morning you eat, it comes back up. It comes up not only to respond to the nutrients, but also to take those nutrients in and break them down into energy and building blocks. Or it sometimes will even decide I've had too much, and it'll then send signals to try to make you realize you're satiated. And then that gets very complex. You get into very complex mechanisms that are already known about satiation. But it's not a just on or off. It's very closely regulated by insulin levels, for example, and glucose levels. In cancer, there will be some mutations that will alter that flux or that those change in levels of activity, so that it will be chronically on, maybe at just a low level, but the chronic activation, which is no longer regulated, will contribute to improper regulation of that cell's growth.
Yeah, that makes sense. So it's under natural circumstances, you expect the system to be waxing and waning according to all of the other sort of rhythms of life that are happening on circadian timescales and things like that. Like in a normal natural situation, you would really never expect TOR to sort of always be on at some chronically elevated level.
Right, unless you're eating too much or
Yeah,
not exercising enough, all that stuff. Yeah.
Okay, and then so inching towards the study that you guys did, I know that as you said, sort of, there's this patchwork of how complete our knowledge is of the different sort of aspects of TOR and how it works. We know more about how certain nutrients activate and regulate the system than others, and there's been sort of some blind spots here. We know about certain amino acids that affect TOR. I'm sure we know a lot about glucose and sugars, but it sounds like, based on my reading of your paper, that we one of the blind spots here has really been how fatty acids and particular fatty acids tie into TOR and affect the TOR system. So can you give me just like a basic sense of like, what are the nutrients where we have a pretty solid understanding for how they plug into the TOR system, and what were some of the blind spots here as it relates to dietary fats?
Sure. So I think as you hinted at before, Piers, and as John explained, so I think the mechanisms for how TOR is activated by amino acid availability and also glucose availability is fairly well established, and I think we certainly have a clear picture of the exact proteins and the pathways that are involved in that regulation. And actually, there's a lot of crosstalk between the core regulatory mechanisms for these different nutrients. But I think fundamentally we have a very clear idea of how glucose and certain amino acids at least activate TOR. Yeah, when it comes to fats and lipids as a whole, we think that's been a bit of a blind spot in all this. I think some contributing factors, when you think of lipids as an entire kind of class of nutrients, they're extremely diverse.
Right.
They have different cellular localization patterns, different functions as well. So I think within that class as a whole, you have all these different offshoots about how each lipid class or subclass could regulate cell function. The specific class of nutrients that we were curious to study were these essential fatty acids. So as the name suggests, these are fats that you can only obtain from dietary sources. And as I alluded to, even when you consider the incredible diversity of lipid species, there are really only two fatty acids that are defined as essential. And these are the omega-6 polyunsaturated fat linoleic acid and the omega-3 fat linolenic acid. And it's really only these two that it's widely accepted. The body can't synthesize them endogenously, and you have to get them from the diet. So I think when we think about omega-6 fats and linoleic acid in particular, that's mostly obtained from dairy products, meat, seed oils, and they're largely associated with pro-inflammatory processes. So linoleic acid is the main precursor for arachidonic acid and prostaglandins as a whole that drive inflammation.
And so just to give people a concrete sense there, so prostaglandins, they are lipid-derived mediators of inflammation. So they're basically little pieces of fat that come from things like the omega-6s, and they turn inflammation on, essentially. And those are the things that and non-steroidal anti-inflammatories inhibit. So that's probably something people have heard of.
Exactly. Aspirin as well. Yeah, yep. Things like that. Exactly. And it's really interesting just to add to that, because prostaglandins as a whole they have a wide range of both autocrine and paracrine effects. So they can signal back to the cell that's producing them directly to affect growth and proliferation. And then in a cancer context, they have a really remarkable ability to just remodel the microenvironment as a whole.
Hmm.
So this can lead, when we think about the immune system, this can lead to actually immunosuppression, and can inhibit, for instance, the response of cytotoxic T cells to a tumor. Um, when we look at the second essential fatty acid, so the omega-3 fat linolenic acid, this is largely obtained from kind of leafy greens, flaxseed oil, and also from oily fish as well. And it's widely accepted that these are anti-inflammatory. So what's really interesting about these two fatty acids is that their conversion to their downstream metabolites use the same enzymes.
Hmm.
So
Yes.
you can envision kind of a picture now where both of these fats are competing
Yeah.
against one another for the same enzyme.
Yeah.
Um, and these omega-3 fats are largely associated with resolving inflammation.
Hmm.
So this is where, especially when we think about these fats, I think the idea of dietary ratios and balance is very key.
Yeah, I'm glad you brought that up because I did want to get to this for people. Things that I think are important there. So you mentioned they're competing for access to the same enzymes. So automatically, this gets you thinking, okay, proportions are going to matter. And when we look historically, my understanding is that pretty much any traditional culture where we have any data to suggest it, not even just traditional as an ancient cultures, but basically before, 100 years ago, people were getting, roughly speaking, at least, about a 1-to-1 ratio of these two things. And when you understand, I think a key thing that you mentioned, John, is, you know, the omega sixes tend to create or be the substrate for the creation of pro-inflammatory stuff, and then the omega threes anti-inflammatory stuff. The thing that I didn't know originally, and I think a lot of people miss, is at least with these forms of inflammation, it's not just inflammation kicks up and then it just sort of like runs out. It's actively regulated on the on and off side. And so having a kind of balance here makes a lot of sense. Is that how you see it?
Yeah, it's definitely the balance is definitely important. I mean, it's not like turning off inflammation completely. You always want to have some.
Hmm.
Your body needs to respond to different challenges, and so you want to be able to have some inflammatory response, or you want to have an anti-inflammatory response. So in the old days, when it was 1-to-1 or 2-to-1, that balance is really great.
Hmm.
Um, Unfortunately, in today's not climate, today's dietary exposures, the ratios can be 10, 20, 30-to-1 of
Hmm.
omega-6 fatty acids to omega-3. And that's where you really disrupt the balance. And
Yeah.
that's where you really promote an inflammatory response that is unhealthy. And
Yeah.
again, can be a healthy inflammatory response, but too much of it is unhealthy.
Right. Right.
And it's really key is that balance.
Yeah, and in principle, being too high or too low on either side of this equation would be bad. Like if you had no omega sixes at all, you would probably struggle in kicking off inflammation when it was needed, like when you were injured or when you had an infection. And obviously that would be an issue, just as being omega-3 deficient, you wouldn't have anything on the anti-inflammatory side, and you obviously don't want to be deficient in that. But in practice, the modern environment has a lot of these omega-6s, as you said, and way more than the omega-3s. And so there's just this sort of bias embedded in the modern environment that's skewing the ratio to have high levels of omega-6s compared to the omega-3s. And so that's skewing us towards the inflammatory side of this whole equation.
Yeah, and the balance goes the other way too.
Mm.
If you have too much omega-3,
Hmm.
and there are consequences there, like some cardiotoxicity issues and
Hmm.
things like that. So, but it's unlikely that you're going to be omega-3 exposed in with the
Yeah.
diets we have now.
Yeah. It's unlikely you'll be omega-3 overexposed. It's also very, very unlikely that you'll be deficient in omega-6s. You'd have to, that would be an
Yeah.
amazing if you could find someone who could even pull that off somehow. Yeah. So you don't, you want to balance here, but so the absolute levels and the ratios probably matter, I would imagine.
Yes.
Exactly.
So let's go over, you mentioned this a little bit. Omega-6 intake is higher than it's ever been. It can be 10-to-1, 20-to-1, 30-to-1 routinely. I've even seen estimates that, depending on how you count it, the average Western person, the average American is getting 10%, maybe even 20% of their calories from linoleic acid alone, which is kind of astounding if you think about it. One
Yeah.
molecule is that much of people's diets.
Exactly. Yeah. Yeah.
And so let's talk about linoleic acid in particular. So this is the most common omega-6 polyunsaturated fatty acid. It's found in high levels in lots of processed foods. Probably the most famous one and the highest concentration dose is going to be seed oils. You guys looked at things like safflower and sunflower oil, but all the major seed oils have high levels of linoleic acid. What do we know about linoleic acid from a biochemistry perspective? What are the major properties of this molecule, and what's relevant here to know?
Sure. So I think in terms of its kind of biochemical properties, so as we alluded to before, it really is the main precursor for the cell to synthesize downstream omega-6 fats that kind of drive the inflammatory processes. So the cells can make arachidonic acid, for instance, which is the main precursor of prostaglandins from linoleic acid. I think it's really interesting to also touch on, it's a great point that you raised before that, at its core, it's an essential nutrient. So if you're deficient in it, which is unlikely, but it could still happen, this has, serious physiological effects too. So I think linoleic acid deficiency is associated with kind of flaky skin. So the skin that more easily blisters, for instance. It leads to brittle hair. So there's a lot of consequences as well of being totally deficient in it. Um, but yeah, the fact that it's mostly derived from these types of components that are becoming way more common in Western style diets as a whole, and the fact that again a substantial amount of our calories coming from this one nutrient, I think it's something that we need to pay close attention to.
Mm-hmm, mm-hmm. And yeah, the other thing that I think maybe is worth mentioning here too is, so when we say essential nutrient, what we mean specifically is the body cannot synthesize it itself. So as
Exactly.
opposed to the monounsaturated and saturated fatty acids, even if you never ate a single molecule of any of those things, your body can make those sort of as needed. But this also, I think, brings us to an important point about regulation and the potential for dysregulation or for things to go out of whack. Is when the body has the ability to synthesize something like, say, a saturated fatty acid, whether or not you're giving it exogenous saturated fatty acids, there's a lot of regulation that it can tune things up, tune things down. But when something has to be consumed, when your body can't create it, like linoleic acid or like the omega-3s, there's just sort of like less regulation there because there's no endogenous mechanism to produce it. And so in general, in my experience, in my read of the literature, that means you're more prone to be able to overconsume such a nutrient. Is that how you think about it?
Yeah,
Yeah.
yeah.
All right. Well, what else should we talk about in terms of background here? So you've got the omega-3s, the omega-6s. Omega-6s generally, like linoleic acid, very, very prominent in our food supply today. They are the precursors to a variety of inflammatory mediators like the prostaglandins, the leukotrienes, and other things. Omega-3s generally are on the resolving side of inflammation. ALA and other things, they tend to be found in other types of foods. Fish oil is essentially omega-3s as opposed to omega-6s. Um, one thing I wanted to ask you guys about before I forget. So linoleic acid in particular, we're going to be talking about your study and inflammation and stuff like that. What happens when linoleic acid is oxidized? What is it about this chemical that allows it to be oxidized, and what generally happens when a molecule like this gets oxidized?
Sure. So I think so typically a lipid or fatty acid's propensity to be oxidized as a whole is closely linked to how saturated or desaturated it is. So, when we say polyunsaturated fatty acids, that mostly refers to the number of double bonds that are in at a chemistry level, the carbon chain of a lipid or fat. For instance, when you think of linoleic acid, this has two double bonds. When you think of arachidonic acid, it has four double bonds.
Mm-hmm.
And the same also applies to omega-3s as well. I think when you think about oxidation at a practical level with how these nutrients are obtained, I think it's interesting from the omega-3 standpoint, this can have major effects on, for instance, the stability of the fat and the lipid as a whole. So I think it's interesting that, if these just omega-3 supplements, for instance, or the pure fat in its form, if it's stored improperly, you will get oxidation of these bonds that would destabilize the molecule and its biological effects would be significantly altered. But I think at its core level as well, these oxidation events are important for the downstream conversion then of these starting fats to their more bioactive mediators.
Hmm. And so the sort of two buckets in my mind when I think about fat oxidation in our cells in animals' cells are there's enzymatic oxidation. So sometimes your cells want to oxidize fats to use them to create energy ultimately, and that's an enzymatic process. That means it's regulated. The cell knows what it's doing, so to speak, if we anthropomorphize it. But then fatty acids like this, PUFAs, can be oxidized in sort of an unregulated fashion. And that's when we talk about lipid peroxidation. And there you can sort of get, it's unregulated. So you can't control the spread of the fire, so to speak. And this can produce toxic byproducts. Is that accurate?
Exactly. So I think when we think about how like the regulated cell form of oxidation, so beta oxidation, for instance, so
Mhm.
how the cell can obtain energy from existing fatty acid and lipid storages. As far as I know, I think the canonical kind of fatty acids that go into that are um, are those that are released from lipid droplets and triglyceride storage, for instance. So these can include like saturated fats like palmitate, for instance, that can then be transported to the mitochondria and oxidized, and then you can generate energy from that. And it's a good point that you bring up. So I think when we look at oxidation of PUFAs as they happen in the cell, these can often damage cell membranes. They can lead to reactive oxygen species. I think there's interesting links now about how these PUFAs in particular might lead to ferroptosis, for instance.
Mhm.
So yeah, I think at the cellular level, that type of oxidation as well can have profound effects.
Mhm. And so let's start talking about your study now. So we know we've got triple negative breast cancer. It's a big problem because this one's difficult to treat. We know you guys mentioned this is actually becoming more common in younger populations. So that's doubly alarming. We know that there's probably some kind of dietary modulation, like to all cancers at some level. We know about the mTOR pathway and its connection to anabolic growth. This obviously ties into cancer because cancer is a form of unregulated growth. It's an anabolic thing, I guess. But on the other hand, you guys mentioned, there's all of these sort of mysteries still to be explained in terms of where these cancers come from, how they tie into diet, and how all of the different specific components of diet, including fatty acids like these that we've mentioned, tie into the mTOR pathway and all of this stuff. So how did you bring us to your paper now? How did you get to this paper in terms of why were you looking at fatty acids like linoleic acid? Was it just because they're more common in the diet? Did you have some other reason to start investigating these particular nutrients?
Yeah, I guess I can start with that. So I think the reason why we were interested in these essential fatty acids to begin with is just because of kind of the gap that we thought existed in the mTOR literature about how these nutrients could potentially regulate the pathway, and in particular, what components or what cellular proteins are involved in or could be involved in sensing these nutrients. Um, so I think for us, the first experiment that really got this whole idea off the ground was it was very simple at its core. We just took a collection of breast cancer cell lines that kind of mirrored the clinical heterogeneity of the disease. So it was cell lines that either had estrogen receptor expression, HER2 expression, or combinations of progesterone and all combinations essentially, and then also triple negative cell lines.
Mhm.
And the first question we asked was very simple at its core was, okay, how do these cell lines respond to omega-6 supplementation? And we looked at a broad range of phenotypes, but what was really consistent is that the triple negative breast cancer models seem to respond profoundly to linoleic acid
Hmm.
supplementation. So even as basic, when we looked at 2D proliferation, these cells could use these fats to enhance their proliferation. When we looked at 3D models, so using spheroids, for instance, linoleic acid was able to drive the growth of these 3D spheres only in the triple negative models. And I think the initial result that really kind of pointed us to the direction that mTOR might be regulated in different contexts was at the signaling level. So when we treated these different breast cancer models with linoleic acid, we saw that mTOR activity could only be switched on in the triple negative breast cancer
Hmm.
models.
Hmm.
So I think these kind of collective phenotypes and then the signaling observations really led us to think that okay, there's something unique about this subtype of breast cancer. that's making it able to utilize this specific nutrient.
Yeah, and that's, I think that's one of the important things to emphasize here is, you guys really dug into some of the details and showed how the different breast cancer types respond in different ways to these nutrients. And one of the things I think that's very important to mention here is, you mentioned in your paper how there's been these conflicting results in the literature. Some of these clinical studies find a link between diet and breast cancer, or this part of diet and breast cancer, and some of them don't, or some of them find the opposite. And it's very confusing. And there's two things I want to say about that. One, when you have that much in the literature and it's conflicting, right? You can people cherry pick this and that from the literature all the time, and you can kind of concoct any story you want around it. But two, this brings us to what I think is a fundamental issue, which is even in, when we look at clinical studies, which sometimes, people really look to first, or they only look at, you have to be very, very, very careful, even if they have a big sample size, and even if, it's a gold standard trial, whatever, because if you don't subtype the different cancers, if you're not looking at genetic differences between people, if you're not really parsing all of their dietary differences and all that stuff, there might not, the signal might not be telling you much of anything. And I think that was one of the lessons I took away from what you guys did.
Yeah, yeah, you definitely hit the nail on the head with that. And I think we can look to see how, even across like drug clinical trials, looking at drug efficacies, how they can maybe be designed better. But I think when we look at the nutrition trials specifically, it's as we were alluding to, I think, even one source of why some studies might be conflicting is something as fundamental as how fat intake was recorded.
Right.
So some studies, for instance, use dietary questionnaires. So
Mm-hmm.
it requires patients or people in the trial to be rigorous about, not only what did they eat today, but maybe also a week ago or 2 weeks ago.
Yeah. Yeah.
You compare that with studies that are, actually take blood draws and look
Right.
at serum levels. There are some that are even more rigorous, where they might take a fat biopsy, for instance, and then do metabolomics on that tissue, then to actually have a readout of fat intake over an extended period of time. So it seems like nutrition studies as a whole, you get very much this mixed bag of the protocols that were used to measure fat intake and things like that. Yeah.
Yeah, yeah. So walk us through some of the first experiments you did, some of this in vitro work. You basically, you guys can give cells different amounts of each of these fats, omega-6s, omega-3s. I think you looked at linoleic acid, so that's the most common omega-6, as well as ALA, which is one of the more common omega-3s that people hear about. So you've got an omega-6, you got an omega-3. When you gave them the cells and you measured TOR activity and all that stuff, what are some of the highlights here in terms of what you found?
So yeah, I think the main takeaway that we found, at least from these initial experiments, was that omega-6 linoleic acid, or LA, in the triple negative models was a potent activator of mTOR signaling. And interestingly, what we saw with the omega-3 ALA essential fat is that it either had no stimulatory effect, or it could also modestly but still significantly reduce mTOR signaling even below its kind of baseline starve conditions.
Hmm.
I should say that this is an active area of interest for our research at the moment, to understand maybe now the omega-3 signaling effects. But I think the dichotomy between both the omega-6s and omega-3s was very clear at the phenotypic and then signaling level. And then I think, once we really established those clear phenotypes and signaling effects, we were curious about what biomarkers now might be dictating these responses. So because it's clear that triple negative subtypes, rather than hormone receptor positives, have these clear effects, we looked at publicly available data sets. So for instance, from the TCGA, from the MetaBrick study, for instance, which looked at genetically characterizing very extensively more than 2,000 primary human breast cancers. And when we did this comparison between triple negative and receptor positive tumors, one of the most highly overexpressed genes in the triple negative subtypes was this lipid chaperone, FABP5, or fatty acid binding protein 5. And when that came up as one of the top hits, that really caught our attention, because certainly within the last 5, 10 years or so, this lipid chaperone in particular has been linked to certain cancer types. So, for instance, prostate cancer. And it's also, from its biochemical function, it's one of the lipid chaperones that has the highest affinity for omega-6 fats and
Mhm.
linoleic acid. So
And what
normally,
what is a
yeah,
chaperone protein like this doing at a very basic level?
yeah, sure. So so as a whole, the family of FABPs or these lipid binding proteins, there's 10 members in
Hmm.
total. Um, their kind of broad biological function is to bind lipids and fatty acids. They might be in the serum or in the blood circulation. They could also be intracellular, but it's really to bind these lipids and transport them across cell membranes, for instance, to other tissue types in the blood.
Hmm.
And then even when you're in the cell, these chaperones play a role in localizing different fatty acids. So you could get them to the mitochondria, for instance,
Hmm. Hmm.
for beta oxidation.
I see.
I think there's some interesting studies, especially with FABP5, showing that it can transport lipids to the nucleus
Hmm.
and affect transcription in that way. So
Okay,
those
so
are kind of yeah fundamental biological functions.
we're talking about dietary fats, linoleic acid. It's a fat molecule. We are basically just a big bag of aqueous solution. So it's actually quite a challenge to think about, well, how does the body move around these fatty things that don't like moving in water? And one part of the answer to that is we have these things called chaperone proteins of all different types, and they move fats around. They move them from one tissue to another through the bloodstream. They can get them from the outside to the inside of the cell. And basically, you were saying you started to see these hints that linoleic acid was driving mTOR, and then you did some bioinformatics work basically, and you saw this
Exactly.
gene kind of light up in that analysis, and it happened to be this chaperone protein that just happened to be one that was really associated with linoleic acid.
Exactly. So I think it's important to note that, out of, there's 10 family members in this FABP kind of protein family. And it kind of each class or each member has an affinity for different types of
Yeah,
fats. So
yeah.
as we hinted to before, they're extremely diverse. You can have different carbon chain length, you can
Hmm.
have different numbers of double bonds, things like that. So there is quite a degree of promiscuity between each lipid class or
Yeah.
chaperone class. But for the large part, each has a preferential binding partner, so to speak. And for
Yeah.
FAB5, it's these omega-6 PUFAs, these polyunsaturated fats, and then specifically
Hmm.
omega-6s. Yeah.
So hypothetically, that would mean that if you had really, really abnormally high levels of FABP5, you would get more omega 6s in than you otherwise would into the cell.
So I think yeah, it's an interesting point. So I think the way we've been thinking about it is kind of calling it a perfect storm of events. So if you have high levels of FABP5 and maybe also a very high intake of linoleic acid from your diet, this could create kind of a scenario then where cells across the body and if you have a specific cancer type as a whole, the availability of these nutrients might be enhanced. Yeah.
Mm-hmm. Mm-hmm. And so the question of the link to tour here, how direct is it? When you started doing these experiments, you might imagine, okay, linoleic acid is making some of your tour signals go up in your assays. Maybe it's getting in the cell, but then other stuff is happening, and it's like an indirect effect. Versus linoleic acid is literally coming in and touching the mTOR protein and directly affecting it. What did you guys find there? How direct was the effect between linoleic acid and mTOR?
I think before we get to that, I think it's really interesting to think about what Nikos has said. He picked up this data
Mm-hmm.
from bioinformatically, and then we asked, okay, that's very cool. We have FABP5 is elevated in many cancers, and it tends to be high in triple negative breast cancers. But then he actually did the experiments and looked for FABP5 expression
Mm,
in the hormone responsive versus the triple negative.
I
And what
see.
was very cool was that triple negative had high levels of FABP5 and the hormone responsive cancers did not. And then when he knocked it down using molecular biology techniques to reduce the expression of FABP5, those triple negative cancers no longer responded to omega-6. And when he took those hormone responsive cancers that had very low levels of FABP5, and he overexpressed the protein in those tumor cells, now they responded to omega-6 fatty acids.
Wow.
So that alone, and there was a lot of more, there were many, many more experiments that were done to support that. But that basic observation really lit the fire to try to understand how that FABP5 protein when bound to omega-6 fatty acids, was then activating TOR.
Yeah.
And Nikos can tell you more about the mechanistic aspects of
Yeah.
it. I want to say also, we started off with, okay, we have these diets, Western diets, let's say,
Hmm.
Western diet, the Mediterranean diets. And there are many things that are associated with Western diets, like high sugar, high omega-6 fatty acids, and so on. But then the the field in nutrition really needs to do what Nikos has done, is to take these complex mixtures and start breaking them down and trying to define how individual components of seed oil or
Yeah.
individual components
Yeah.
of the Western diet can affect. And then that leads us to what I just discussed. And then we want to understand molecularly at a biochemical level how that then activates TOR.
Yeah.
And there's reasons for doing that because it then reveals possible therapeutic targets. And then Nikos can tell you more about the molecular mechanism of
Yeah.
what he discovered.
Yeah. Okay. So basically, in other words, you had these triple negative breast cancer cells. You had other non-triple negative breast cancer cells. The triple negative ones happen to have strong expression, high expression of this FABP5 protein, the chaperone that's good at grabbing omega-6s and bringing them in the cell. So you're like, wow, that's interesting. But then John went in and said, okay, well, if that's really key here, you should be able to take that away from these triple negative cells, and then you won't see some of this, or you'll see it less. And you, in fact, did that. And then you actually then went and increased FABP5 in the other breast cancer cell lines that don't normally have it highly, and that rendered them more sensitive to omega-6s. And
Exactly.
that starts to get you towards an explanation for why, like, okay, there could be a dietary effect for people with this type of breast cancer, but not that one, because cells can behave in these kinds of different ways.
Exactly. Yep.
Okay. Okay. Interesting. And so, yeah. So, sort of what happens next in the story, John?
I'll let Nikos tell you.
So yeah,
Sorry,
so I
sorry,
think
Nikos. Yeah.
so I think in terms of then the specific mechanism, so I think one clue that came to us about this was, so there were a couple of factors. So I think the first idea was the time frame from which this omega-6 linoleic acid could activate TOR.
Mm-hmm.
And in all of our experiments, we saw robust activation, in the span of minutes. And I think that 30 minutes is when this led to the most robust activation of TOR. And I think that was an important clue because, as we mentioned, linoleic acid is an essential nutrient. It could be converted to these prostaglandins as well, and it can maybe feed back and have some indirect effects on the cell that might then indirectly activate TOR. But when we did some other kind of metabolic assays and other experiments, we saw that at least within this time frame, within 30 minutes, those other kind of maybe indirect effects weren't really changing.
Mm-hmm.
It
Mm-hmm.
was really specific to this kind of fairly acute activation of TOR. So then that led us to think that maybe there might be some kind of direct activation or direct interaction between FABP5 and TOR. And this was informed by kind of two main findings. So in the literature, it has been shown that FABP5 is really unique in that when it's bound to linoleic acid specifically,
Yeah.
it can translocate to the nucleus and directly interact with transcription factors,
I see.
like PPAR-gamma, for instance. So it's been shown in prostate cancer to regulate metastatic phenotypes, for
Mm-hmm.
instance.
Mm-hmm.
And it's really interesting because this is, as far as I know, the only FABP family member that's been shown to have a direct interaction with another protein
I
in
see.
response to it binding linoleic acid.
So this isn't something that's just sort of like grabbing the linoleic acid and throwing it into the cell, and that's sort of the end of it. There's a complex that actually goes all the way into the nucleus and it's starting to turn on and turn off genes and do things like this.
Exactly, in response to this specific nutrient.
Yeah.
Um, and then I think the other piece of evidence that kind of caught our attention is, work from John's lab several years ago was instrumental in defining how TOR interacts with its direct substrates. And it's really through this amino acid sequence called a TOS motif. So many, certainly a few direct mTOR binding partners and substrates have this TOS motif that's defined by a highly conserved phenylalanine residue and then alternating kind of hydrophilic and hydrophobic fatty amino acids. And when we looked at the amino acid sequence for FABP5, we actually saw that at its end terminus, it had a very promising candidate sequence that when we compared this motif to established Tor interacting partners, looked fairly similar. And so that was kind of the start of us thinking about, okay, could FABP5, when it's bound to linoleic acid, directly interact with mTOR? And I think all the experiments, all the mechanistic experiments that we did subsequently, I think we were very careful to make sure that, the interactions between TOR and FABP5 were occurring when we did immunoprecipitations, also immunofluorescence, things like that. And really, this interaction, though, only occurred when linoleic acid was present.
Mm.
So it seemed to kind of set the scene for defining a very specific context through which this nutrient activates TOR under specific conditions.
Wow. And then, so one way to start thinking about some of this stuff so someone like me who doesn't know a lot about the cancer side, but knows more about the inflammation side, I might think to myself, okay, you guys can give these cells linoleic acid. It can get into the cells. It's having this effect on this key metabolic pathway on mTOR. Maybe this is like, okay, the omega-6s come in and then they get chopped up into prostaglandins and the inflammatory stuff they're sort of famous for. And then that stuff is pushing us in the mTOR cancer direction. Or an alternative could be like, no, it's linoleic acid coming in and sort of directly plugging into mTOR. It sounds like it's really more of the latter, right? Linoleic acid is coming in and directly plugging in here.
I think both is happening.
Mm.
I think as you increase the amount of linoleic acid in your body, you're going to increase the amount of TOR, but some of it's also going to go to producing these inflammatory prostaglandins, and cyclins, and again, it's all about balance. But I think the more and more that you get in, the more and more you're going to activate TOR, the more you're going to produce these inflammatory cytokines, prostaglandins. I'm sorry, and I think in biology it's never one or the other. It's always shifting the equilibrium.
Yeah.
And we basically have shifted the equilibrium so that we're going to get both path processes occurring, and that's probably deadly.
Yeah, yeah. Is there any reason to think that there could be some kind of double whammy effect here where, okay, you have a direct effect on mTOR, but also if you have more inflammation in parallel, would that potentially feed in to the cancer side of this directly, or is this like, okay, you have this cancer effect via mTOR, and then you could have more inflammation as well, and that's just bad because you don't want that anyway, or do these things possibly tie in together directly?
So I think we have in our paper we did do some cell biology experiments to at least try to address this. So in these triple negative models that we treated with omega-6 linoleic acid, we were curious about how different signaling nodes, so some pathways that are directly regulated by TOR, others that are regulated by these pro-inflammatory processes could be affected. So I think what was really interesting is, so when we treat cells with linoleic acid as a whole and check general signaling profiles, we see that there's TOR-specific activation of specific substrates, and also other substrates that are linked to pro-inflammatory processes. So
Mm-hmm.
we see activation of AKT, for instance. We see increased phosphorylation of ERK. And the key experiment, though, is in these models, when we knock down FABP5, we were able to kind of parse out which nodes were being regulated. So I think what was very interesting is in cells that were depleted of FABP5 and then that were stimulated with linoleic acid, we saw that these pro-inflammatory signaling nodes, so AKT, ERK, could still be activated, which maybe suggested to us that, okay, there's certainly a pool of linoleic acid that can be converted to arachidonic acid, prostaglandins, and then can feed back. Whereas the activating effects on mTORC1 specifically was lost. So this kind of pointed to us that these signaling events are they're driven by kind of pools.
Mm-hmm.
So when a cell has linoleic acid, there's going to be a subset of that total linoleic acid pool that's activating TOR through FABP5, and then you're going to have another pool that's being converted to arachidonic acid
Right,
and
right.
prostaglandins and then feeding back. So this is where the idea of kind of like this perfect storm is coming into play. Yeah.
Yeah, yeah. And Yeah, I'm going to step back a little bit. So when we think about, well, actually, before I do that, so what would you say? Oh, there's actually one piece I forgot about completely. So you did the experiments we've talked about already. You then looked in human tissue. What did
Mhm.
you find there? Let's do that next.
Yeah, so I think for these experiments, we had access to two types of samples. One were these patient-derived xenograft tumors that we were able to profile that were either triple negative or receptor positive. And when we looked at kind of tumor intrinsic levels of FABP5 and total omega-6 fat content, we saw that triple negative tumors seem to have higher levels of both of these components compared to receptor positive.
So you saw these
And
signals in human tissue that mirrored all of the stuff that you saw in the wet lab.
exactly. And then I think perhaps the most striking results that we saw were in patients. So, by collaborating with oncologists here at Cornell, we were able to get access to serum from patients that were newly diagnosed with either triple negative or receptor positive disease. And the serum was collected right before their first surgery. So, this kind of provided a very interesting snapshot of what their metabolic profiles might be looking like, what
Yeah, yeah.
might look like. And again, what we saw is in patients with triple negative disease, serum levels, so now kind of systemic circulatory levels of FABP5, and interestingly, omega-6 fats were higher in these triple negative patients
Hmm.
compared to receptor positive patients,
Wow. So triple negative breast cancer patients seem to have literally more omega sixes floating around.
and FABP5 as well.
Yeah,
Yeah.
yeah.
And
And so
I think it's important that we mention that the FABP5 levels are elevated doesn't necessarily mean that their intake of omega-6 fatty acids is also elevated. Probably what we could say is yes, they probably do have higher levels of omega-6 fatty acids in their diet, but because they have more FABP5, it's soaking it up.
yeah, they're
And,
effectively more
and
sensitive to it in this respect.
Mhm.
exactly right.
Wow.
And then generating a situation where you have higher levels of omega-6 bound FABP5. And I think the other thing that comes out of this is really, which is very important, is that this told us that FABP5 would be a really great biomarker for
Right.
determining whether we should whether this patient is being affected by omega-6 and also whether this patient should be treated with therapies that are targeting this process.
Right, right. Yeah, I mean, up to including, if we're essentially talking about linoleic acid sensitivity here, right away that would tell you a dietary strategy you could take.
Yeah. Yeah.
Yeah, yeah.
Interesting, interesting. So let's talk about cause and effect here. Is simply elevating linoleic acid in the diet of a rodent, say, is that sufficient to have an effect on triple negative breast cancer?
Yeah, so I think it's an important question and a bit of a loaded one too. So I think there's two answers. So the studies that we did were looking at the effects of a high omega-6 rich diet on an animal that already has triple negative breast cancer.
Hmm.
So in the studies that we did in the paper, animals were injected with triple negative breast cancer cells. They developed these mammary tumors, and then we put them on these omega-6 or omega-3 rich diet. And we saw that once the tumor had already formed, its growth was potentiated by a high
I see.
omega-6 rich diet.
So
Um,
increasing
yeah,
omega sixes in an animal that already has triple negative breast cancer basically makes it worse.
exactly. I think kind of the second part of the question is at least from our perspective, it's less known and could be even more controversial to say whether, if you're cancer free and then you were to suddenly have high amounts of omega-6 linoleic acid, is that tumorigenic or carcinogenic in any way? That from our study, we're in no position to kind of comment on that, and I think that would need
Hmm.
a lot of work to
Hmm.
dissect. But at least when a triple negative tumor is present, and then you're kind of adding on high omega-6 levels, at least in our animal models, that seemed to
Hmm.
drive growth.
Hmm.
Yeah.
Two things that came up for me when I was reading this paper and talking to you guys that connect to other work that's been done recently that I've had discussions with the podcast on that I would love your take on are one, as we mentioned earlier in our discussion, colon cancer is another one of these cancers that we're seeing creep up more and more in younger and younger people, which, that starts to make you think like, okay, this could be an exposure thing from early life onward. I've talked with Yeatman Halliday from University of South Florida, and they've got some work basically showing that colon cancer can be driven by this inflammatory state of lipid dysregulation that ties into the omega-6s as well. Kind of evocative in some ways of some of the things I'm hearing here. And the other one is I had a gentleman from the UK on, and he has I'll just give the headline result. I'm not sure if you guys know this work, but basically he showed that aspirin, which we mentioned is what inhibits the prostaglandins, which is they come from the omega-6s. Aspirin actually inhibited metastasis of certain tumors in, I forget what type of cancer, but I don't know. There's some interesting threads that seem like they start to coalesce here around how these omega-6s tie into both inflammation and metabolic dysregulation and in some cases even metastasis. Stepping back and just thinking about the general literature here, what comes up for you guys, and how are you thinking about these omega-6s in a more broad context?
Yeah, I can start with this. So I think the way that we're thinking about it is, so actually, I do want to start by saying that when we think about nutrition in a cancer context, I think fundamentally the way we think about it needs to be in the same way that we think about drugs and targeted therapy. So when we think about breast cancer, for instance, we know there are all these different subtypes. It's common sense that you would treat one subtype with a specific drug.
Yes.
Simply because we know the genetic markers, we know what biomarkers to look for, and we have a clear idea of how patients will respond. We need to apply that same rigor when we think about nutrition. So kind of maybe these blanket recommendations of eat this nutrient or cut out this fat completely, it might not be the right advice for a certain patient or a certain
Right.
group of patients. And to your question now about thinking about the literature and what's out there, I think it's a really exciting time because there are these kind of pieces of evidence that are out there that's saying, okay, certain patients or certain groups or certain populations are responding differently to these nutritional interventions. And really, how can we now link this into clear guidance for the right group of patients? I think what's really interesting, and it's something that we're discussing more frequently, is when we look at omega-6 fat metabolism, there are these interesting population studies looking at polymorphism, so genetic variants in the enzymes
Yes.
that are converting linoleic acid to arachidonic acid and pro-inflammatory prostaglandins. And what's really interesting is that there are these genetic variants in certain patient populations, so those mostly from Hispanic or African ancestry, that actually makes these enzymes more active and
Yeah.
more efficiently able to convert linoleic acid to these pro-inflammatory mediators.
Right.
Now,
Right.
I think it's really noteworthy because with a lot of these studies, the previous way of thinking was that it's minimal conversion. So you
Right.
can have as much linoleic acid as you want, and you're not getting prostaglandins or arachidonic
Hmm.
acid. That might be true for certain patient populations, but certainly for others that might have these
Yeah.
variants, the advice needs to be tailored.
Yeah.
So I think these pieces are out there. I think our FABP5 story adds an element to this, and I think what will be really exciting is now putting all these together to have kind of a defined nutritional
Yeah.
advice.
Yeah. I was so glad you brought up some of that because I was actually gonna to try and pull us in that direction. So some of the enzymes involved in fatty acid metabolism of the omega-6s and the other things, as you mentioned, sorry, the name's throwing me off because it says
Yeah.
John right below you, and so every time I look at you, I see John's name. But one of the aspects of the human genome where you see the most diversity is actually in fatty acid metabolism genes, like these FADS genes. And as you mentioned, Nikos, people are very different in terms of this is my interpretation of the literature. I'm just going to say that to make it clear for people, essentially in their linoleic acid sensitivity in the sense of how readily do they convert it to these downstream inflammatory components. And to me, the interpretation is very clear. That has very different dietary ramifications for different people. There should not be a blanket. There's no magic number that's going to be a one size fits all. People's
Exactly.
sensitivity effectively to the potential, the bad effects of some of these nutrients can vary quite widely. And as you guys did in your study, you looked at triple negative breast cancer as opposed to other ones, and you looked at a specific nutrient. You really parsed things out and checked things very carefully. And One of my frustrations with the clinical literature, and I know this is hard, it's not like I'm not necessarily a criticism in that sense, but you can't do a full genome sequence for everyone. You can't measure every marker for everyone. I get it. But sometimes we get very confused when we don't parse the data and bucket people in these different ways because it obscures what is in essence what we're talking about is personalized medicine. Someone could be really sensitive to something that someone else is relatively insensitive to, and all of these things. And I think that's really important to keep in mind.
Exactly. Yeah.
Yeah, I think that gets to the importance of understanding at a molecular level how these things work. Because, it was basically trying to understand the mechanism of how omega-6 works that led us to FABP5. And so now we can add FABP5 to that list of biomarkers that will dictate how people will respond to it. In addition to where it's expressed, knowing where it's expressed may be also very important because it may not just, it's likely not going to only affect cancer cells, it's likely going to affect other cell types as well. And so we just need to know where FABP5 is and how its expression is regulated.
Yeah. And so linoleic acid was what was driving mTOR and a lot of the effects here. And just to remind people, and if there's any other detail here that we should add, let's get into it a little bit. You also looked at the omega-3 ALA, and so it did not show the same effects, but can you just sort of remind us what exactly was ALA doing in some of these assays?
Yeah, so I think at a high level, so if, for instance, it didn't seem to induce proliferation in the triple negative cell lines in the same way that omega-6 LA did. Um, it had no stimulatory effect on any of the signaling pathways we were looking at. And even in some instances, when we were doing like these time courses, it may have even reduced mTOR signaling even below its kind of baseline starve levels. So overall, it just seemed to have a suppressive effect on many of these phenotypes. The mechanism is something that we're really interested in dissecting more, but one interesting piece of data that we included in the paper was when we treated triple negative cell lines that again have high levels of FABP5 with ALA, so this omega-3 fat, it did seem to reduce protein stability. So in cells treated with this ALA fat, total protein levels of FABP5 seem to reduce over time.
Hmm.
Again, we don't really know the exact mechanism for it, but this suggests to us that it might be having an effect on protein stability. And this could provide at least one explanation for why
Hmm.
omega-3 ALA was having this kind of
Mhm.
blanket suppressive effect.
So you've got your omega-6 linoleic acid, you got your omega-3 ALA, and this is another example where they have distinct, if not somewhat opposing effects on biology.
Exactly. Yeah.
Hmm. Yeah, it is kind of remarkable to me how these two arms of lipid biology, they really do seem to be evolved to oppose and sort of counterbalance each other.
Yeah, they're in direct competition. Yeah.
Yeah, literally. And again,
Yeah.
so you said that earlier, the enzymes that process them are some of the same enzymes. And so if you've got more of one, it's going to crowd out the other ones.
Exactly.
Yeah.
Yeah.
Yeah. So what do you think some of the high level takeaways? We've already discussed some of this, but it's good to get some repetition in. All things considered, the specific experiments you did that we talked about, as well as some of the sort of bigger picture stuff about how diet ties into general health and cancer in particular. What are some of the major takeaways here in that you guys have taken away in terms of how people should think about things like dietary fat and seed oils and omega-6s in particular?
You want me to take it? Yeah.
Yeah,
Yeah.
sure.
So I think the biggest takeaway message for us is that context is key. Um, I think when we need to apply the same rigor to nutrition research, and specifically if we want to integrate nutrition in a cancer patient's journey, we need to approach it with the same rigor that we do when we think about drugs and small molecules, and that is at the molecular level and at the biomarker level, identifying which patients are going to respond to specific nutritional interventions because it's certainly not a blanket recommendation that'll work for everyone. And we've seen this in the case of just comparing different breast cancer subtypes. But as we discussed before, when you look at patients that are otherwise healthy but have these inherent variations that differ in their sensitivity to linoleic acid intake, for instance, I think that just really highlights that we need to better understand which patient populations are going to respond to specific nutritional interventions, and the context is key to define this.
So obviously, your study was looking at all the things that we talked about in the context of this triple negative breast cancer. But given how common linoleic acid is in our modern food environment, and as we said, some people, it's like 10-20% plus of their calories are coming from this one molecule. It's very unusual that something would be that concentrated in an animal's diet. Given that fact, and given the molecular biology that you uncovered, that linoleic acid is plugging into the mTOR system this way, do you think it's likely that the effects that you've seen here are limited to triple negative breast cancer, or do you think that there's good reason to suspect that high levels of linoleic acid might have other consequences for other diseases as well, other cancers in particular?
Yeah, I think that Nikos has a little bit of data on prostate cancer as well. The more aggressive prostate cancers also are responsive to omega-6 fatty acids. And we just haven't looked at, broadly at other cancers. But my guess is that a lot of the intestinal cancers, cancers of the intestinal tract, pancreatic, colon, and so on, may also be sensitive and and in that case, we also need to look at FABP5 levels. And again, I just want to say not all will be the same. It really will depend on this biomarker. FABP.
Yeah. So again, this is the chaperone protein. So people that just naturally have higher levels of this thing are going to be effectively more sensitive to this cancer promoting effect.
And we don't know actually if high intake of omega-6 fatty acids induces the expression or stabilizes protein.
Interesting.
So there can be this sort of positive feed loop that is negative in terms of cancer growth.
Yeah. Interesting.
It could help. And also there are factors that may be secreted by adipose tissue. So if you're obese, it may be that they produce more FABP5.
Right.
And so there are all these things that this study has revealed, all these questions that we need now to address, because I think this is going to be much bigger than just triple negative breast cancer, which is big enough as it is, but it'll be much greater.
So I think you kind of just hinted at it, but what's on the horizon for either or both of you in terms of work that's going to come from this study? What are you guys looking at right now?
Yeah, so I think one area that we're interested in is kind of the role of this signaling axis, so FABP5, mTOR, in other chronic diseases. So as John mentioned, adipose tissue and fat cells also have high protein levels of FABP5. So one thing that we're really interested in is, okay, if you have high intake of omega-6 linoleic acid, is this driving certain inflammatory phenotypes in fat tissue?
Yeah.
And I think, that's kind of a fundamental question that we have, but I think it has knock-on effects to other areas. So we're also curious about how diet influences what's known as kind of like unhealthy aging and these inflammaging phenotypes, which is like, this chronic low-level systemic inflammation, and studying the relevance of FABP5 in these processes. So I think we're really excited to look at the effects on other chronic diseases. Yeah.
Mm-hmm. What's your take on, so obviously at some level, everyone cares about nutrition. We all eat food. Some people really, really care about nutrition, and they get into it. A positive side effect of that is that a lot of people really get interested in nutrition science and related elements of science. It's actually a great way to get people interested in science, generally speaking, and to start caring about evidence. And, one of the things I just want to point out and compliment you guys on here, especially for certain elements of my audience, is, just how closely you check everything. Like, when we're talking about experiments like this, you mentioned, we saw this cool bioinformatics association, but then you went and checked, and then you went and turned things up and turned things down. You really sort of really functionally check everything. And you can do that in the wet lab in certain ways that you can't do it in the clinic, granted, but, just really important to always check things. But a negative side effect of people getting into nutrition science and all that stuff is they often really get, it's hard for people to maintain a dispassionate sort of ideal of science perspective on this stuff. And they get really tribal and they get really opinionated on stuff. And one of the areas where people get really opinionated all across the board, educated, uneducated, professors, regular, everyone, is the seed oil stuff. Because, on the one hand, we've got this whole history of seed oils being called heart healthy by the most prominent institutions in the universe. And on the other hand, you've got people saying seed oils are terrible, they're inflammatory, that you should avoid them at all costs, and everything in between. One of the tough things in those debates that I experience is, people will be arguing back and forth about this stuff. People will say, we understand the biochemistry here. The omega-6s, there's a straight line you can draw from omega-6s to these inflammatory mediators. Like, it's very crystal clear, and the biology is fairly well worked out at the biochemical, cell biological level. And then other people with a more clinical orientation will say, okay, but why don't I see any randomized control trials where these serum markers of inflammation go up when we give people more omega-6s? Given some of the things we talked about, how you have to parse data between different types of patients with different predispositions and all of that stuff, what is your take on why there aren't more clinical studies showing a clear connection between omega-6 dietary intake of omega-6s and inflammation?
I think that there can be more clinical studies now that we made some of these discoveries, like FABP5 and its binding to omega-6 fatty acids. So I don't think that the tools were available before to answer those kind of questions in the clinical trial. But now they are, and I think that future clinical trials should take this information and clearly should be gathering samples to measure this FABP5, omega-6, and other nutrient components. I think one of the problems with the arguments of Western diets versus Mediterranean diets, or seed oils versus not seed oils, is that most of the arguments are not fact-based. And it's really important to understand the mechanisms, to understand the real facts of how these things work. And then we can start dissecting what's good and what's bad in these complex mixtures that we're having. I mean, seed oils, not only omega-6, but depends on what the source of the seeds are. If it's corn oil, were they grown on farms that use pesticides? And, it's so complex. And that's why we need to really break down all the components and really understand molecularly how those components work. And then I think that will guide how the clinical trials are designed.
And I think, just to add on to that, when we think about how future nutritional studies and trials are done, I think we need to think about having a diversity of patients and different patient populations that are included in these trials. So, we spoke earlier about these polymorphisms and genetic variants in genes that regulate omega-6 metabolism. These might occur in people or in populations that might be traditionally underrepresented in certain trials and nutritional trials as a whole. So I think what might happen is, when we go back to analyzing these data, maybe it's not a representative sample of actually the human population as a whole and the variants that might be present. So I think, especially from the fat debate, future nutritional studies should really consider this and think about their inclusion criteria and exclusion criteria, things like that. Yeah.
Mm-hmm, mm-hmm. Yeah, there's some fascinating work there. And the fact that you said that is triggering me to, there's some really good work from Robert Reich and others in the sort of human population genetics world. And there's a pretty compelling story right now that, not only is there variation in things like fatty acid metabolism, but a lot of that was tied to like the advent of agriculture and stuff. When people started farming more and they started ingesting more arachidonic acid, surprise, surprise, it affected how the genes involved in that metabolism changed. And when you start to step back and think about the different populations and stuff, what you just said, Nikos, makes a lot of sense. It could very well be that just those people that are sort of have the highest linoleic acid tolerance tend to be the ones included in a lot of these studies more often, and that will naturally skew what is found or not found.
Exactly. And again, that's and when we think about dietary recommendations, that's not necessarily a problem for that specific population.
Right.
But then when we get into the nuances, then in the specific context of then why this variation occurs, this advice might need to be tailored accordingly. Yeah.
Final question I'll ask you guys. answer as much or as little as you want, but a more personal question, given what you're studying in this paper and in the world that you guys live in, and all the literature that you've been studying over the years, how has all this stuff affected your own relationship with omega-6 fatty acids and related compounds?
I can start. I definitely take supplements, omega-3 supplements, fish oil supplements. I definitely watch what I eat. I don't even remember the last time I went to a fast food restaurant. It's been a long, long time. So I'm very aware of what's out there, and I try to manage it. We do other things in the lab that have revealed other nutrients and other vitamins that are important. So I actually use that information to supplement my diet. And I think the other thing for me personally is not only the diet, but again, it's been mentioned before, but maintaining some level of exercise. One thing that's nice about New York City is we're always walking, always walking in the city. So that helps in that area. Sleeping, eating good diets, all those things are important. So on a personal level, the science that we're doing, I believe in very much, and then I let it dictate how I respond to it in terms of my diet and my physical
So you're
activity.
not cooking with safflower oil at home, I take
I
it?
don't cook with tap oil. I don't use bacon grease.
Yeah.
Yeah, yeah.
At least for me, I'm pretty much exactly the same. So I try to avoid processed food, fast foods as much as possible. Unfortunately, I do love dairy and meat too much, so
Yeah.
I do try to limit intake. But where possible, I think the main takeaway message is balance.
Yeah.
So when I think of what I'm eating or what I'm cooking for dinner, I try to have balance
Mhm.
as for.
Actually, one thing I wanted to ask you about, this is kind of a detail, but I did want to ask it because it's on page 1 here in the graphical abstract. So this story is basically about linoleic acid, a dietary omega-6, and what happens, too much linoleic acid is basically what we're talking about here, and we're talking about breast cancer. And you've got a cartoon which is meant to represent the Western diet, which I assume here is meant to represent very high levels of linoleic acid. And there are two components of this cartoon. One is safflower oil, and one of them is meat, and it looks very much like a beef steak. Now, I am confused about one of those two being there. Can you explain the choice there?
Yeah, so I think when we think about meat, what's an important question, and I think John touched on it a bit earlier, is kind of how that meat is raised and how it's how the cattle is raised. So I think there's what's becoming appreciated is there's differences in linoleic acid content and arachidonic acid content of meat products, whether the animal was kind of grass fed or grain fed for its entire life. So I think it's an interesting consideration, but also an important one because it does kind of emphasize the trickle down effect of, even at the earliest stages of agriculture and food production, the kind of the knock-on effects on how this can affect us. Yeah.
Yeah.
So the meat is there because of how to highlight the differences between maybe grass-fed and grain-fed animals. Yeah.
And
Yeah.
even in the case of grass-fed animals, when they're brought in before they're sacrificed, they're put on grain to fatten up. And so that's how they really accumulate a lot of omega-6 during that period. I'm not sure how long it is, a month or two or so.
Yeah, you're talking about
But
grass-fed, grain-finished, as opposed to
yeah,
pure
Yeah.
grass-fed.
Yeah.
yes,
Yeah.
yes, exactly.
Yeah, I
Exactly.
think one thing worth mentioning here for people is, yeah, I mean, so just to echo what you said, Nikos, yeah, like if we're talking about seed oils, sometimes you're not eating a seed oil, but you're eating something that was eating seed oils
Yeah,
because that's how it
exactly.
was
Yeah,
raised, and that affects its fat profile. But
exactly.
also differences between the animals. So, like cows, for example, are very much they're affected differently by the fat content of what they eat based on their metabolism than say a pig. So if you feed a pig grain, for example, it can pretty much just have seed oils as its fat. And
Mm.
so sometimes lard and things like that can have very, very high levels of omega-6 content, which people don't often realize.
Exactly.
Right.
Yeah. Yeah.
And that's even true for chickens.
Yeah,
They're
chickens too.
corn.
Mhm.
Yeah,
And so, yeah, we have to watch out what we're eating.
yeah, yeah. Any final thoughts? Anything you want to reiterate before we sign off here?
I don't know, at least from my end, again, context is key.
I probably you shouldn't record this part, but I think that what's really important is that this science on mTOR started in the late 80s. And I got a grant in the early 90s, and it's been continuously funded all these years. And again, we've gained all this information that has led us to this point. But I think what's important for me is to make sure that the readers understand that this is not just an overnight discovery. This is a discovery that takes decades and decades of research that's supported by the NIH. And I'm a little bit worried right now about how what the funding situation is going to be like in the future. And it's very important that readers understand that these things take a long time, but the amount of information and the types of therapy that have really been developed over the last 15 years is amazing. We're making such amazing progress in all areas of metabolic disease, not just cancer, but obesity, diabetes, cardiac disease, and so on. And that it's really, really important to support the basic science to get us to then allow us to translate it and then go into clinical trials and then finally into people.
Yeah. And just to riff on what you're saying there, John, it's not only been 40 years of research. This is stuff that started in yeast, and they didn't know. No one knew that they were going to discover mTOR exactly. And it was going to lead to this.
Yeah, exactly. Yeah.
All right. Well, thank you guys very much for your time. Very cool stuff, and I look forward to following what you do next.
Thank you for having us.
Yeah. Thank you very much.
Visit mindandmatter.substack.com to find all of my content. If you like to read like I do, then you should check out the ReadWise app. It's totally free to download, and if you go through the link in the episode description or on my support page, you can get a free 60-day trial for zero cost. ReadWise is a really cool app that allows you to import digital highlights from over 30 different platforms such as Kindle, iBooks, PDFs, and more. It makes it really easy for you to review and find and share excerpts and quotes from whatever it is that you're reading, whether it's PDFs of scientific papers or books that you're reading on your Kindle. And you can even save and use highlights from physical books. I read a lot of physical books before bed, for example, or when I'm traveling. And the ReadWise app actually integrates with the camera on your phone so that you can take a photo of a page that you're reading, highlight a specific chunk of text from that page, and export it to the app so that you can reference it or find it or share it later. They even allow you to export really cool looking graphics of some of the highlights that you have in the books or the papers you're reading so that you can share them in an engaging way. So check out the ReadWise app today if you are a reader and you like to reference and organize what you read. Go through the link in the episode description or on my support page or in my free weekly newsletter at mindandmatter.substack.com and try the ReadWise app 60 days free today.